Can someone? Oh, okay. Yes. Okay. Good. All right. So my name is Hokuto Kazama. I'm working here at RIKEN CBS, and so the instruction today was that I should give you an overall introduction to the field of olfaction in the first hour or so, and then in the next hour, introduce some of our work done in my lab. So I will follow that instruction. And I'd be happy to answer any questions. So please feel free to ask any questions and interrupt me, okay? Right, so olfaction. So people may not appreciate too much the importance of the sense of the smell compared to the sense of seeing, for example. However, I would like to start by saying how much we rely, or animals rely, on the sense of smell. Okay. So the first question is, how is a sense of smell useful? Is it useful at all? What are the functions of the sense of the smell? So apparently, some animals heavily rely on olfaction to find a mating partner. And this is critical for reproduction. For example, here I have shown an image of a male silk moth, which has an exquisite sensitivity to a female pheromone. And you can probably see a huge antenna here. This is the antenna. This is much bigger than the typical insects that you find out there. And they will show zigzagging behavior and then the looping behavior to finally arrive at the position of the female. So it's important for this animal and other animals as well. Another thing that we also did many hundred years ago, probably, is to forage for food. So foraging means navigating towards a food and finding a food source. You may not be able to directly see with your eyes the place of the food. However, with the sense of the smell, you may be able to identify that, oh, there must be an orange somewhere. So I will go towards that position and so on. And foraging is, again, critical for us to obtain energy. But biting on an orange is not always good, because once it goes bad, you want to avoid being food poisoned. So this is another function that we are using daily to avoid intaking harmful substances. And on a more positive side, we appreciate food, not just because of the taste of the sense of taste, but also the sense of smell. So basically, in order to form a proper flavor perception and enjoy the flavor of the food, we need olfaction. When you just smell an orange, for example, you use an orthonasal olfaction. However, when you eat an orange, then you will simultaneously recruit gustation and also retronasal olfaction. So the odor will come back to the nose. And it is the combination of these two senses, and of course, plus the somatosensation, the crunchiness also matters. You use vision and so on. So flavor perception is a truly multi-sensory perception. But the retronasal olfaction is a key factor. So you should thank your nose for being able to appreciate the food that you have every day. Okay, so I hope that you have recognized that the nose is constantly functioning. And before going into the details of the circuits, I would like to give you the basic structure of the olfactory system. So the olfaction starts by sniffing odorous substance, and the odorants will arrive at olfactory epithelium, where the olfactory sensory neurons reside. And then the signal is propagated already into the brain. It's very quick. And the primary olfactory center is called the olfactory bulb. And then the processed signal will reach already olfactory cortex. So with just 2 synapses, you can reach the cortex. This is very shallow compared to other modalities, which makes it easier to understand the cortical processing. Now, when I blow up this region, it looks like this. So this is the blow up of the olfactory epithelium in your nose. And the odorants will interact with the cilia of the olfactory sensory neurons. And many olfactory receptors are expressed on the cilia. Okay? And here is the mucus. So the odorant will bind and activate olfactory sensory neuron. And then OSN, or olfactory receptor neurons, will send signal to the olfactory bulb. You can see other types of cells. One is called the supporting cell. The other one is called the basal cells. Basal cells are stem cells, and they give rise to olfactory sensory neurons because olfactory sensory neurons live for only 30 days to 60 days or so. So they are constantly being replaced by the new ones. The right picture is a microscopic EM image of the epithelium, and O represents the olfactory sensory neuron, and you can see the cilia here. Okay. Now, after the olfactory sensory neuron activation, they send the axons into the olfactory bulb, the primary olfactory center. And they can do that because even though there is a bone, so of course you have a bone to separate your nose and the brain, but there are holes on the bone. And this bone is called the cribriform plate, but there are small, tiny holes made, and through that hole, the axons can reach a specific, roundish structure called glomerulus. There are no needs to remember the specific terms today, but perhaps it's nice to remember this one term, glomerulus. The plural is glomeruli, which acts as fundamental information coding channels in the olfactory circuit. So the olfactory sensory neurons arrive and form the glomerulus, and then they synapse onto the second-order neurons called the mitral cells and the tufted cells, and they send signals to the olfactory cortex. So this is the basic structure. Okay, so next I would like to move on to this question, which probably you have thought about once in a while, which is how can we perceive a variety of different odors? We encounter many different types of odors, and even when, let's say, when you travel abroad and then smell a completely novel odorant for the first time, you can still sense, right? So that means our system must be able to perceive a huge variety of different odors. So how can we achieve that? And I would like to introduce to you some molecular mechanisms that underlie this ability to perceive a lot of different odors. So a breakthrough was made by a pair of researchers. One is Dr. Richard Axel, and the other is Dr. Linda Buck. So Linda was a postdoc in Richard's lab. Richard's lab is situated in Columbia University in the US. And they both were together were awarded the Nobel Prize in 2004 for their discoveries of odorant receptors and the organization of the olfactory system. So basically, they have discovered a large family of odorant receptors. This is one of the first papers published in Cell in 1991. The title is interesting. A novel multi-gene family may encode odorant receptors, a molecular basis for odor recognition. So I cannot imagine myself publishing a paper with a title having may. And then this can be published in Cell, and then this led to the Nobel Prize. But research is always like that at the very beginning. When you find something very novel, no one knows about it. And I think it's a good attitude to be conservative and say, oh, it may encode, rather than being too confident and, oh, this encodes definitely the odorant receptor. So yeah, even though Richard Axel and Linda Buck are very strong person with confidence, but I learned that they started from this paper. So olfactory receptor neurons, again expressed on the cilia of the olfactory receptor neurons, are 7 transmembrane proteins, and it is G protein coupled. So when an odorant binds the olfactory receptor, it activates the G protein, and the G alpha olf then activates adenyl cyclase that produces cyclic NP, and then that will open the cyclic nucleotide gated channels, and it will pass the cations like calcium and sodium. And that is the reason why the olfactory receptor neurons can be depolarized. Okay. So I started off this section by saying that we can perceive a variety of odorants. So the question is, how many of those receptors do we have? This is a really nice study by Nimura-san, who counted the number of olfactory receptors in different animals. Well, let's start from humans. We have 396 intact olfactory receptors, so the red one. But you notice that we do have a similar number of pseudo-olfactory receptors, 425. So pseudo means it's not functional anymore. The gene is there, but it's not functional. So this may tell us that the mutation can happen quite rapidly in these genes. But anyway, there are 400 or so, and this is quite large when you compare to the number of genes that the photoreceptors use. There are only 4, right? RGB plus the ROD receptor. So compared to that, you have 100 times more odorant receptors. And given that about 20,000 genes encode proteins in human genome, we utilize about 2%. We dedicate 2% of the genome to just encode olfactory receptors. So it's a lot of, it's a big percentage. However, mouse has much, much more, like 1,130 intact olfactory receptors. And the largest known is elephant. They have about 2,000. I don't know why elephant had to have this much receptors, but this is a fact, interesting fact. Also dogs have 800 or so. But anyway, there are a large number of odorant receptors. And the trick is that a single olfactory receptor does not recognize just one odorant. It can recognize multiple odorants. And from a different point of view, an individual odorants can be recognized by multiple receptors. Okay, so for example, this odorant is recognized by these 2 receptors, and so on and so on. So you can use a combination of receptors to encode a single odorant. And this gives an astronomical number. Okay, so this idea was proposed by Linda Buck in 1999. Now, with that, we would like to move on to the details of the circuits. So how are signals processed in the brain after being recognized by the receptors? Well, so odorant receptors are expressed in olfactory receptor neurons in the nose. And the first general rule that is applicable to many, many animals is so-called the one neuron, one receptor rule. So each olfactory receptor neuron expresses only one type of receptors. So for example, this blue ORN expresses only this type of receptor, the orange one, another single type of receptor, and so on. This is conserved from flies that I'm going to talk about in the next section, and to the humans. Now, the next general rule that is found is one receptor, one glomerulus rule. So olfactory receptor neurons expressing the same receptor converges to the same glomerulus. I'm sorry about this. Okay. So what do I mean by that? So let's focus on this orange olfactory receptor neurons. They express the same receptor, and they target exactly the same glomerulus, one and the same glomerulus. So when a neuron is expressing another receptor, they converge to a different single glomerulus, and so on and so on. So this way, information about the activation of particular receptor is not intermingled in the primary olfactory center. Okay, so how did the people prove this? Well, Peter Mombers, then at Richard Axel's lab, made a mouse where olfactory receptor P2 type can be visualized like this. So in this particular mouse, only the P2 OR expressing olfactory receptor neurons are labeled in blue. And here is the olfactory epithelium. And then if you follow the axon, it converges to a single glomerulus. This is a very beautiful image. When I first saw it, I couldn't believe that this can be happening in my nose. But well, this is mice, but not human nose. But and a single pair of target glomerulus is actually being labeled by this blue marker. So there are about 2,000 glomeruli in mice, but only a one pair is labeled. So with the power of genetics, they were able to find this rule. Now, so signals have arrived at individual glomeruli. And then after that, they send signals to the second order projection neurons called mitral and tufted cells in mammals. But before that, multiple local neurons interconnect the glomeruli, and many inhibitory and excitatory interaction happens. And the only final output is being transmitted by the mitral tufted cells to the high order brain regions. Well, there are many brain regions, but here I am showing two example major targets. One is piriform cortex, and the other is amygdala, particularly the cortical amygdala. Well, in the amygdala, you can find a stereotypical connectivity, which I'm going to talk about in more detail in the following slides. But this region is thought to be responsible for assessing the innate valence of odors or innate value of odors, whether the odor is pleasant or unpleasant. That's called valence. Whereas the same mitral tufted cells bifurcate and send axons also to the piriform cortex. And in the piriform cortex, you can find random connectivity. And this brain region is thought to be important for olfactory association, so basically learning and memory. So again, we would like to see some of the original papers that together found out that pattern of connectivity. So the first paper is from Richard Axel's lab. So they utilized a mouse where a single glomerulus expressing mouse OR 1749 is expressed. This glomerulus was labeled in green. And then using this fluorescence, they injected a red fluorescent dye like this, okay? And this dye has a property to propagate along the axons all the way to the end. So if you now look on to the right image, the top is the before injection, or the control brain, I would say, perhaps. Oh, no, it's the same brain, but the labeling of the basic structure. And on top of that, you can see red signals originating from a single glomerulus. So mitral tufted cells in a single glomerulus innervates such a wide region. And you can see that this is the border of the preform cortex. So it innervates the preform cortex and also the amygdala, olfactory tubercle, and so on. And just to summarize this paper in one sentence, projections to the preform cortex is disperse and homogeneous. Well, it's rather disperse and homogeneous, while those to the cortical amygdala are broad. Well, we cannot say broad just from this single example, but it's patchy. It occupies one patch of the cortical amygdala and stereotyped. If you look across multiple mice, you see similar projection patterns. So there are, the projection pattern is different between the preform cortex and the cortical amygdala. Now, another lab called RIKEN lab used an opposite strategy to delineate the circuit. So I'm saying opposite because they did not start from the glomerulus, but they started from a higher order neuron, for example, a neuron in the preform cortex. And then they utilized the rabies virus, which can propagate retrogradely through a synapse. Some of you may have already learned about this, but it can hop one synapse and then label the presynaptic neurons. So the idea is to, okay, let's start from the preform cortex and then figure out which are the glomeruli that are connected to this particular preform cortical neurons, okay? So by repeating this experiment, you can, they reached basically a similar conclusion as the other lab. If you look into the preform cortex, the cortical neurons receive inputs from a variety of glomeruli in the olfactory bulb without any strong patterns. So it probably supports the random projections by the mitral tufted cells of the preform cortex. Whereas if you look at the amygdala, they receive input preferentially from the dorsal glomeruli because it's reddish here, right? So it seems that the projection is non-random. And these glomeruli include those that are sensitive to the oxyurine, which is aversive for mice. And it makes sense that the amygdala receives those inputs because that brain region is thought to be important for processing innate value of odors. Okay, so I would like to introduce yet another paper, which was published just 1 year after those two papers from Kensaku Mori's lab at the University of Tokyo. So Igarashi-san, the first author, wanted to understand the relationship between the odor responses of the neuron and the structure of that particular neuron. So he did a heroic experiment, which must be very hard to conduct, which is summarized in panel A. Okay, so he first did optical imaging of odor responses, and then roughly figure out which brain regions respond to a particular odor that he prepared. Okay, and then he inserted an electrode, a glass electrode, into that region, and then made a juxtacellular single cell recording. So juxtacellular recording means you attach a glass electrode onto the membrane of a soma of one particular neuron, and then record extracellular spikes from that neuron. So you're confident that you are recording from a single neuron, unlike tetros or silicon probes that are not completely attached next to the soma. Okay, so then after doing that, he can introduce a tracer into that single neuron, and then wait for 3 days for the tracer to be transported all the way to the edge of the axon, and then visualize the tracer and 3D reconstructed the entire structure. And the result is shown on the right image. So he has recorded from a single mitral cell, in this case here, the soma is here. And then he found out that the single neuron extends its axons throughout a large brain areas, including a preform cortex and the entorhinal cortex, olfactory tubercle, and so on and so on. So this gave him a different access to the different type of information, which were not accessible by just doing anatomical studies. So from that, he has made this summary cartoon showing that the mitral cells innervate a larger brain regions compared to the tufted cells. And then also he labeled mitral cells as slow cells and tufted cells as fast cells, because the response onset to the odor was much faster for tufted cells, and the mitral cells started responding to odors much later. So there were not only anatomical differences, but also functional differences between these two major types of second-order neurons. Okay. And this gives you a little bit more detailed circuitry of the mammalian olfactory circuit and beyond. So starting from the glomerulus, I told you about the mitral and tufted cells, and they innervate the preform cortex, cortical amygdala, anterior olfactory nucleus, but also entorhinal cortex, and so on. So from there, of course, the signal goes to the hippocampus, from amygdala, hypothalamus, and then so the sense of olfaction does not go through thalamus, but eventually it goes to the thalamus. And also frontal cortex, orbitofrontal cortex, and so on. There is another set of organ called vomeronasal organs in rodents, especially well studied in rodents. And the mitral cells in the vomeronasal organs respond to pheromones and so on. And they have a differential innervation to a particular part of the amygdala. So that's the difference between the accessory olfactory bulb and the main olfactory bulb. Okay, so far so good. All right, so I'm going to move on to the next section, which is about the computations. So how is odor information encoded in the brain? I'm going to introduce about 4 different types of computations, starting from a spatial code. So spatial code means that the olfactory information is encoded in a spatial activity pattern. So this is an example study done by Katz's lab in 1999, using an intrinsic imaging targeting the rat olfactory bulb. So intrinsic imaging is a relatively simple imaging method, which follows the oxygen level in the cortical area. Its temporal resolution is not so high, so it's not a perfect method, but it's a relatively nice method to look at the wide region of the brain area. So with that method, this group has applied 2 different odorants in this case, and then found that different odors, octanol and amyl acetate, recruit a different set of glomeruli. So in response to octanol, these 3 glomeruli shown in arrows and arrowheads are recruited. But with amyl acetate, additional glomeruli labeled by black arrowhead is also recruited. So from this, they concluded that individual odors activate a different combination of glomeruli. Okay? So you can use the space to encode particular type of information. Okay, so this is simple. The second one is also relatively straightforward, which is a rate code. Odor information is encoded in a firing rate of neurons. Here I'm introducing a work done in Nao Uchida's lab at Harvard, where they trained the rats to do an odor discrimination task. So the rat will start by sniffing a particular odor in an odor port, and then depending on the identity of the odor, rats should choose the left port or the right port, and if the rat is correct, they can get a water reward. Okay? And in panel B, you can see the sequence of experiment. So at one point, the odor port opens and the rat starts sniffing. You can see that the respiration level frequency increases in anticipation because the odor is not applied yet. Okay, so the odor is applied here. So you can tell that this rat is well trained already. And then depending on the choice, the water is delivered. Okay, and then they recorded from the second order micro-tufted cells in the rat olfactory medium. And the spikes, extracellular spikes recorded are shown on the top right. They have, in this case, applied 4 different odors, A, B, C, D, and the blank air control. Okay. And each black tick mark represents a spike. Okay. And the spikes are shown with respect to the time of inhalation onset. So the inhalation onset is time 0. Okay. And then because they have gone through multiple trials, you have many rows. Okay. So that's how you read this figure. And then when they calculated the average firing rate over 160 milliseconds and then plotted just the firing rate, they obtained this kind of data and then found out that the odor D evoked a stronger response. So they said that, okay, from the firing rate, you can extract information about odor. Okay? So odor D has come. And of course, this is one way to encode information employed by many neurons, in many brain regions. But if you look more carefully, the temporal code is simultaneously being employed in this brain region. So odor information is encoded in the dynamics of firing. So to look at the data more carefully, they did not use a large bin size, which was 160 milliseconds, but this time they used a much smaller bin size, 20 milliseconds. And then they started to find a difference between a response to odor A and odor C. So the firing rate was quite similar and even similar to the blank. However, if you look at the spiking pattern, it was different. The dynamics was different. Okay. So the blank and odor B is kind of similar, but A and C is different, even though the firing rate is similar. Okay. So this is evidence that the temporal code is being utilized in the olfactory system. Okay. All right, then the next one is called the primacy code, extracting the early responses to achieve concentration invariant odor identification. So concentration is very important to recognize the smell. So if the concentration is, for example, very high, some odors will be aversive to us, even though it is attractive at low concentration. So odor, not only odor identity, but odor concentration needs to be recognized by the brain. So how does the brain do that? So Dmitry Rimberg and others have came up with this hypothesis, the primacy code. And I'm going to explain that to you using this slide carefully. Okay, so on the top, I am showing the activity in multiple different glomeruli in the olfactory bulb of a rat. Okay, and then in the middle row, they are showing an odor concentration over time. So here the concentration increases gradually. Okay, and in the bottom row, they are showing the glomeruli, the identity of glomeruli that are activated by this odor concentration pair. Okay, so the glomerulus I was recruited first, and then glomerulus D, and then later glomerulus Q. This was the sequence. Okay. Now they increase the concentration of the odor a little bit higher. Okay, so this axis is the odor concentration, and people have found out that if you increase the concentration, a larger number of glomeruli are typically recruited. Okay, so now you can see that on top of glomeruli D, I, and Q, 3 additional glomeruli are being recruited. However, the order of recruitment still remained the same. Again, I, D, and Q were recruited first because they are perhaps more sensitive to this particular order. And so on and so on. So even though if you people have these researchers have changed the concentration, they found a similar sequence of glomerular activation. So if there were neurons listening to this sequence of glomerular activity, they can always recognize that this particular odorant has arrived, regardless of the concentration. Okay, so I thought that this was a pretty strong hypothesis, which have been supported by another lab recently. Okay. The text is just a summary that I have explained. All right, so I have talked about 45 minutes or so, and with that, I hope I have given you a rough summary of the olfactory system. And after taking a break, I would like to introduce some of the work done in my lab, which addresses some of the unanswered questions in the field. Okay. So if you have any questions, I'd be happy to take those. Yes.
I have a question on the one receptor one flow. And generally speaking, in order for 2 neurons to meet the same place, there needs to be a guiding molecule. And the diffusion of the guiding molecule is important to the communication between 2 neurons. But in the one receptor one glomerulus flow, there is a very small plate between 2 neurons, and I thought that this plate restricts the diffusion of the light. But so my question is, how are 2 neurons recognize each other?
Yeah, so I guess it depends on the origin of the diffusible guiding molecule. If it's utilized within the brain, then it's fine. You don't need to think about the propagation through the cribriform plate. And I believe that's the case. So the axons are interacting with each other before reaching the glomerulus. So that's one mechanism to bring similar types of olfactory receptor neurons together. And on top of that, there is a guidance molecule that guides to the particular glomeruli. So there are multiple mechanisms happening to form that precise circuit. Any question from the audience or from people online? Oh, it has increased to 34 people.
Sorry, not so important question, but why Rinderback and Axel use the word may novel and in the cell paper, why they use the word may?
May, okay, I see. So at that point, they didn't have evidence that the receptors encoded by those genes indeed respond to odorants. That wasn't, that experiment was not done yet.
They isolate the genes, right?
Yeah.
But how they didn't express and they didn't confirm the receptor and odorant substances.
Yeah, so the they didn't go that far. So that's why they didn't conclude. They felt that the evidence is not there yet.
Yeah, thank you.
Well, expressing the functional olfactory receptor even in vitro is quite difficult, and it's a tricky experiment.
Is it difficult? So how about introduce the HEC to 93, the olfactory genes, receptor genes, and express receptors, and like stimulate with all the
that's the exactly kind of experiments that are done later on by many other researchers. But the stably expressing receptors itself took some time to make it happen. And finding the right expression level is also an issue. It's a good system to understand the basic principle, but what happens in vivo needs to be later confirmed in an intact animal. So, of course, electrophysiology or imaging in an intact animal would give you a more strong evidence. There may be a question here. Okay, I have two questions. The first question is in page 22. The colors in the preform cortex are more than 6. Some patches exhibit different colors or a combination of these colors. Is there any possible explanation? Another question is on page 30. I wonder whether the concentration of odor will cause the change of smell itself. Page 30. Oh, so I should have labeled the page numbers, but maybe I can answer the second question first. Yes, as I have alluded, if you change the concentration drastically, it will change the perception, the olfactory perception. So that's why you have to specify both the identity of the odorant and the concentration of the odor. And the first question, I think, is about Miyamichi et al. This one, I think. Okay. And the question was, there are more than 6 colors. Oh, okay. So 6 colors representing different glomeruli, but here there may be different, more than 6. I don't know whether there are more than 6. Maybe well, so this is a cartoon summarizing the basic, the fundamental result. So I don't know how accurate these cartoons are, but a single neuron in the cortex could receive inputs from multiple glomeruli among these 6. So in this case, maybe the authors utilize a different color. That's just my guess. I need to read the figure legend again. I'm sorry, I don't have a definitive answer. Okay. All right, then let's take like a 5 minutes break, and then let's come back at when? What time is it now? Yeah, maybe 6 o'clock. Okay, right. Yeah. Oh, yeah. Thank you. Okay, let's begin. Okay, good. So I would like to focus on two questions. One is, how are odor objects represented in the brain? And the other is, how does the brain compute the value of odors rather than the identity of odors? Okay, so I want to explain you first what I mean by odor objects. For example, here I have shown you 3 different molecules having different structures. L-menthol, methyl salicylate, and L-carbon. So those are, of course, different substances, but they all smell minty. So we have an ability to perceive that these 3 odors belong to the mint category. And we call that this constitutes a mint odor object. Okay. And this idea can be applied not only to individual odors, but mixtures of odors. Let's say that you went to a curry restaurant and that restaurant was using this particular ratio of multiple spices. And at home, you make curry yourself with different ratio of spices, but still you can perceive this mixture as curry, right? So this is another example that we perceive odor objects and not just individual odors. Okay, I hope that you guys agree that we do not perceive the individual component spices, but the gestalt of curry smell. Okay. So the question is, how does the brain form odor objects in the brain after processing? Okay. There are multiple candidate brain regions, but we hypothesize that maybe the counterpart of the prefrontal cortex, called the mushroom body in insects, could be forming odor objects, because its function is like the prefrontal cortex, to form an olfactory association. So associating a particular odor with reward. And if you want to form an association, the basic computation about odor objects must have been completed by then. Otherwise, it doesn't make sense, right? So that's the rationale behind this hypothesis. And by the way, so I'm going to talk about fruit flies. And here I am showing the fruit fly, Drosophila / vertebrate olfactory circuit. And they are quite similar. So I'm just renaming nose as antenna, olfactory bulb as the antennal lobe, prefrontal cortex as mushroom body, amygdala as lateral horn. And this basically gives you the fly olfactory circuit. And because of this similarity, many researchers believe that we can extract the fundamental principles of olfactory computations by studying the fly circuit. So for example, Richard Axel, he himself has been continuously using both mice and Drosophila. All right, so I want to then explain to you how do we record neural activity from the major neurons called kinome cells in the mushroom body. It's a very tiny animal whose body length is about 2 millimeters or so, and the brain size is much, much smaller. So it must be very hard to record neural activity. So using a couple of pictures, I would like to give you an idea about the experiment that we do daily. So on the right is a female, on the left is a male. These flies are stationary because it's been anesthetized with CO2 coming through a CO2 pad here. Okay, but we want to record in vivo, but we don't want the flies to be flying away. So how do we do that? we came up with an idea of fixing a fly to this kind of a recording plate. This is 3 centimeters in each axis. And this is the top view shows that there is a tiny hole made in the middle. The size of the hole is several hundred microns wide, and that's the size of the fly brain. Okay. If you look from the side, it looks like a stair, as a stair or a step. And we will use a UV curable glue, which can be cured in about 10 seconds or 20 seconds by applying UV light. And to fix the head and the thorax of the fly to this edge. By doing so, because there is a room here for the fly to beat its wings, we can continuously monitor the fly's behavior while applying an odor from below. But because of the hole we made, we can remove the cuticle on top of the head, and using electrodes or microscopes, we can image neural activity. So after fixing the fly to the plate, we bring that plate under the 2-photon microscope in this case, and record neural activity in response to odors delivered by this 16-channel olfactometer. So this is also made in the lab. And using that microscope, we can get an image like this. So this is the fly brain. Maybe Teru has told you about the fly brain. But in this case, we expressed a calcium indicator called GCAM in the second order projection neurons. So they have dendrites in glomeruli in the antelope, the primary olfactory center, and then send axons to the mushroom body and the lateral horn. Okay? So using a genetic line, we can very beautifully and specifically label a particular type of neurons. And by attaching a piezo drive on top of the objective lens, we can move the objective lens very rapidly along the z-axis, along the depth. So with the laser scanning in XY, we can image the entire volume housing 50 glomeruli in flies in about 560 milliseconds or so. So we are cutting 33 optical slices. And then after you image one optical slice, you move the objective lens to next focal plane and then image again, and so on and so on. And by doing that, we can get an image like this in the middle, and the right is the reconstructed cartoon of glomeruli. And I'm going to play a video showing the response of glomeruli to 2 different odors. Okay. And as we expected, a different spatial temporal pattern of glomerular activity can be observed in response to 2 different odors. So this is how we can discriminate different odors. Okay. This is shown in pseudocolor, okay? And then after writing lengthy code, we can automatically register the brain to the template and extract the change in fluorescence in the individual glomeruli. Now, after recording the activity of second order projection neurons, we next recorded the neural activity from the third order canyon cells innervating the mushroom body, the focus of the brain, the target brain region in this section. And again, a genetic line is available that specifically labels all 2,000 canyon cells in this case. And then we successfully recorded from the entire 2,000 canyon cells in response to an odor like this. So having an access to the entirety is the strength of using the simple animal like the fly. This can be very difficult to do even in mice. Okay, so in this experiment, we applied 15 different odors and then recorded the activity of 37 glomeruli. So we couldn't record from all 50 glomeruli, but now we have managed to record from 46 glomeruli. But anyway, response to 37 glomeruli are shown in pseudocolor. So the warmer color means excitation, and the colder color means inhibition. And on the right, are responses of many, many kinome cells, in this case, 320 or so, that have significantly responded to one of the 15 orders that we prepared. Okay? So it's not 2,000. That's because the responses are sparse in the beginning. And secondly, we have applied 15 orders and not millions of orders in this experiment. Well, as you can see, the number of orders eliciting a response is much smaller in kinome cells. While your visual system can automatically detect that the matrix looks more bluish, whereas this matrix looks more greenish or yellowish, suggesting that the responses, order responses, become sparser in the second order processing center. Okay, so those are the raw responses. But because the dimensionality is so high, we wanted to get a rough idea about the type of computation that happens in the mushroom body in a lower dimensional space. And there are many ways to do it, but we utilize a principal component analysis in this case, which is a linear transformation where new axes or principal components best capture the variance of the data. So basically, your raw data is the responses of neuron 1 and neuron 2, or glomerulus 1 and glomerulus 2. But let's say the different orders evoked different activity in these two neurons. And looks like that the best way to capture this distribution is to have an axis like this. Okay, so that will be defined as PC1, and the perpendicular one will be defined as PC2, and so on and so on. Okay. And the data projected to the PC1 axis is called the PC score. And I'm going to show you the PC score in response to 15 different odors in the next slide. Okay, so if we look at PC1, it captures about 40% of the variance. So it captures a decent amount of data variability that we recorded from. And there are 15 different orders. So there are 15 plots here, okay? And so this is the order response projected onto PC1. We can do this for the first top 15 axes, and it looked like this. And here, using a green box, I have isolated an order whose response is significantly different from other orders. So you can visually see that this response to order 15 is very different, far away from other order responses, right? And those responses can be found here and there. And if the responses are different, you can ask the downstream neurons to draw a line between these orders and say, oh, I can discriminate this order 15. Okay, so being able to discriminate is good if your goal is to identify a particular order. Now, if we do the same analysis in the mushroom body, we see quite a different picture. First of all, the percentage of variance explained by the first PC1 is much smaller. And the variance is shared among a larger number of PC. This means that the neurons are more independent of each other in this brain region. Okay? And if you look at the order responses along PCs, now we can find a much larger number of orders that are significantly separated from other orders. So those orders become more linearly separable in the mushroom body as compared to the antennal lobe. So this is one transformation, signal transformation, that is happening in this brain region. To more rigorously quantify this, we trained a linear decoder and then asked whether a particular order can be classified from other 14 orders. Okay? And I'm plotting the classification accuracy in both the antennal lobe and the mushroom body. And for all 15 orders, the classification accuracy was higher in the mushroom body. So this suggests the odor responses are more discriminable in the mushroom. Okay. This makes sense because one of the goals of the olfactory system is to discriminate between different odors. We don't want to mix up different odors. Okay. Now, when we came back to this PC plot, we noticed that not only individual odors, but also groups of odors housed in orange boxes are significantly separated from other odors. And the number of those groups are much larger in the mushroom body as compared to the anthola. So we thought that, oh, maybe mushroom body is more good at categorizing odors than the anthola. Okay, so and this is a population level analysis, and we wanted to look at the responses of individual odors, individual neurons. Sorry. So let's focus on this odor group, comprising odor 9, 11, and 12. Okay. So those odors 9, 11, and 12 are benzonoids. Okay, and we found a Kenyan cell in the mushroom body that are specifically tuned to those 3 odors and not responding at all to other odors. And we found out through analysis that these neurons indeed contribute to population level categorization of these groups of odors. And somewhat surprisingly, we found that group-specific Kenyan cells were observed for various odor groups that do not necessarily share physical-chemical features. So in this case, we initially thought that, oh, these are benzonoids, these are physical-chemically similar. So that's why similar receptors recognize this, and thus eventually a Kenyan cell was able to respond to those 3 odors. But it turns out that even if we picked 3 random odors having very different physical chemical features, we can find Kenyan cells that are exquisitely tuned to those odors. Now, those were individual odors forming an odor object, okay? But I also, in the introductory slide, talked about the mixture odor object. So we tested how odor mixtures are represented in those brain regions. To make a story simple, we prepared only 2 odorants and mixed at different ratios. Okay, so we have 6 different olfactory stimuli. And then first looked at the odor responses in the anthola in a low-dimensional PC space. So x-axis is PC1 and y-axis is PC2, together capturing more than half of the variance, okay? And then starting from the pure 2-methylphenol, if we mixed in the second component, 1-octanol, the responses gradually changed, okay? And then eventually reached to the pure 1-octanol. Oh, this makes sense. You are changing the ratio gradually. So the population odor responses should also change gradually. However, in the mushroom body, we found that as soon as we mixed in the second component, 1-octanol, the response became very, very different. So there was a big jump from stimulus number 1 to stimulus number 2. So that's one observation. The second salient observation was that even if we changed the mixing ratio from 80 to 20 to 20 to 80, so comparing the response to odor number 2 and odor number 5, they were still similar, as if the mushroom body is forming this mixture odor object in a higher dimensional space. Okay? And then going to the pure 1-octanol, the odor responses again changed quite dramatically. So this was a striking demonstration that the high order brain region is doing qualitatively different computation from the earlier brain region. Again, so after looking at the population level responses, we looked at the responses of individual Kenyan cells. Okay? The mixtures are the same. And then we found a Kenyon cell that responded like this. So the first one responded most strongly to pure 2-methylphenol, and the response became smaller and smaller as we decreased the ratio. So this neuron is like encoding 2-methylphenol, and this one is one octanol neuron. Okay, it's easy to imagine seeing this kind of responses. However, besides this, we found a Kenyon cell that specifically and also invariantly, with the same level of response strength, responding to only to mixture and not to the pure components at all. So this is a mixture cell that emerged in the mushroom body. We also found concentration invariant component-specific cells. So this one is invariantly responding to 2-methylphenol, and this one to 1-octanol. So this suggests that even though the olfactory system is not so good at isolating one component, but it is in principle possible to identify a component within a mixture. Okay. All right, I'm gonna move on further to the circuit mechanisms that give rise to those Kenyon cell responses. So how do we form these responses? To address that, I need to introduce to you a little bit more detailed circuitry connecting the antennal lobe and the mushroom body. Okay, so the mushroom body is subdivided into 15 glomeruli, and then the projection neurons make synapses onto Kenyon cells in the mushroom body. Okay, so but the first thing is I need to tell you is that individual Kenyon cells are receiving input from a random combination of glomeruli. Okay? So this has been known experimentally. So let's introduce this mechanism. And also through optogenetics and electrophysiology experiments, we and others have shown that the Kenyon cells perform weighted summation of inputs. So that's another necessary mechanism. And there are GABAergic inhibitory neurons providing feedback to Kenyon cells. So that seems to be necessary as well. And of course, the spiking nonlinearity. So by incorporating those minimal sets of mechanisms that are experimentally observed, we try to simulate the responses of Kenyon cells to different odors. Okay? And this is one example result. All right, so the first step is random selection of glomeruli. And in this case, these random 6 glomeruli were selected. And for example, glomerulus 1 is responding to many, many different odors, very broadly tuned. Glomerulus 2 as well. Glomerulus 3 not responding too much. 5 with some tuning and so on. Okay, and the next step is weighted summation. So we summed these inputs with a particular synaptic weight and then got this response. And by the way, the particular category, odor category that we are focusing on is labeled with orange. So these 3 odors comprise this particular category. At this time point, this response is not specific to these orange odors at all. However, after providing feedback inhibition, you get this kind of differences between odors, and with the spiking nonlinearity, we successfully generate the odor group specific Kenyon cells. So although we do not believe that these are the only mechanisms implemented in the brain, but what this analysis told us is that these 4 mechanisms are sufficient to generate the core responses of Kenyon cells. So how about mixtures? We did the similar thing for mixtures. At the level of glomeruli, none of the glomeruli responded specifically to mixtures. However, after those steps, it's not complete. This Kenyon cell is responding to some extent to pure components, but it is more strongly and invariantly responding to mixtures. This is the grand summary of simulation. On the left are responses to 15 different orders. The left matrix is the actual canyon cell responses observed with 2-photon microscopy. The right is the model canyon cells. Your eyes are pretty good at probably telling that these are quite similar. And similarly for mixtures, we were able to recapitulate the order responses, predict the order responses quite well. Okay, to summarize this first part, we found that at the population level, the category or mixture-specific responses emerges in the mushroom body. And at the single cell level, we found group-selected canyon cells and mixture-selected canyon cells. To summarize the circuit mechanisms, random connectivity, summation of inputs, feedback inhibition, and the spiking nonlinearity were able to generate order category specific or order mixture specific canyon cells. And from here on, a little bit of discussion about further processing downstream. So we have been focusing on canyon cells, 2,000 canyon cells in the mushroom body, but they make synapses onto 34 output neurons. So these are the neurons that send output to further downstream to the association area, multimodal association areas. And the synaptic connection between those canyon cells and the mushroom body output neurons are modified by dopamine neurons. So there are dopamine neurons responding to reward and punishment. And if those two stimuli, the conditioned stimulus and the unconditioned stimulus, coincide, then they typically evoke LTD, long-term synaptic depression at these synapses. And these synaptic changes are thought to be the mechanism underlying learning and memory. And this 3-layered random convergent divergent circuitry can be found in other brain regions in other animals as well. One good example is the 3-layered circuit in the cerebellar cortex in mammals. You might know that the precerebellar neurons send mossy fibers, and the connection between the mossy fibers and the granule cells in the cerebellar cortex is random. And the dimensionality is very, very high in granule cells. Granule cell is the most numerous cells in the human brain. So just like the dimensionality increased from 50 to 2,000 in the mushroom body circuit, the dimensionality rapidly increases. And there are feedback inhibition mediated by Golgi cells and so on. And the climbing fibers provide teaching signals onto the granule cell to Purkinje cell synapses. So the Purkinje cells are the output neurons of the cortical circuit, cerebellar cortical circuits, okay? And Masao Ito, the founder of BSI, and others have found that LTD happens when the signals from the climbing fiber and the parallel fiber coincides. So it's very difficult to image from the entire cerebellar granule cells, but we have shown that with our technique, we can record from the entire 2,000 canyon cells. So our wish is not only just to understand the canonical computations in the olfactory circuits, but to understand canonical computation that can be implemented by these circuit motifs that can be found in other animals. So that's the broader goal that we have. Okay, any questions before going to the last section?
The model of cases and step of weighted summation. How do you decide the weight of each?
Yes. So now the entire connectome is available in flies. So the connectome is the wiring diagram of all the neurons in the brain. and that is determined by electron microscopy. So you can count the number of synapses made between different neurons. So we provide weights based on that connectome. But when we conducted this study, the connectome data was not available yet. And so we looked at the electrophysiology data and then assumed that the weight is some value with some variability. So we implemented some Gaussian distribution to the weight. But now with the new data, we can make a better one.
Yeah, thank you.
Yeah. Okay. Right. So I hope I'm not going too fast. Please stop me if I'm going too quickly. Oh, there is one chat. Is there any possibility of oversaturation, overstimulation for single compound chemicals? I'm just wondering if there are any time-dependent reasons that cause the higher level detection of multi-components. Higher level detection of multi-compounds.
Maybe it's easier if I just speak.
Yes, please.
Okay. Well, so my primary question is, I'm thinking for olfactory detection for humans, if there's a single compound smell, you can become overstimulated to the point that you don't really detect it anymore. But maybe with these hybrid compounds, where we have a different ratio of distribution, the oversaturation doesn't occur. So, but then when I thought about it more, you would think, or this would be detected as some type of decay rate over time, and not an instantaneous kind of lack of detection of single compound alcohols. So.
I see. So to back up a little bit, so you said that if you apply a single odorant at high concentration, one would not be able to recognize that odorant. Is that what you wanted to say?
I'm thinking, well, you would be able to detect it, but I think over time. So I'm thinking of something like smelling gasoline. So it's a little bit different than pure alcohols, or even some smells that can occur in your natural life, some certain odors.
Yeah.
Over time, you don't have a high level acknowledgement of these odors. You become accustomed to them, adapted to them.
Oh, I see. You're talking about adaptation.
Yes.
And your thought is that would
for the pure chemical, or for a single compound, because the constant that I don't know how to say, this compound is pure in its nature. It's kind of fixed, so there's no diffusion between two different chemicals, or between two different odors. So the sensitivity to it decreases, or you adapt to it so that you no longer detect it, or it becomes a discrete abstract object, and you don't pay any. You just the detection of it decays over time. But these hybrid compounds, maybe there's something abstractly related to it, that there's a hint of this other compound, and we have different ratios, of course, and because of that, we're always or this high level memory type of behavior can continuously detect, oh, there's a combination of this and this, so there's hints of this and hints of this. But if it's a pure compound of either type, there's some type of depreciation or decay where it's not detected anymore.
I see.
Does it make sense? I don't really know how to word.
I'm getting what you're trying to say.
Okay, thank you.
So what I think you're trying to say is that in a mixture in a natural environment, different components may be adapted at different rate, so that you might not be able to recognize the gestalt, but you still perceive the components. Whereas
Correct.
if you use a single odorant, that may not happen. Is that the kind of thing that you want
Correct.
to say?
Yeah.
Yeah.
So
Okay.
the Gestalt components, there's almost an emergence of another abstract entity. So we have the single component and we have another single component, but then we have kind of a different behavior, emergent object to represent the combination of these other ones that the depreciation is slower than.
I see. Yeah, it's an interesting hypothesis. Yeah, that may happen. So adaptation happens at various levels. It can happen at the level of receptor, at the level of olfactory receptor neurons, at the level of synapses made between olfactory receptor neurons and secondary neurons, so on and so on. So it's a complex mechanism, but the different odors may have different rate of adaptation. And if so, then the perception may change over time as you hypothesize. So yeah, I think it's an interesting point that could be experimentally tested, actually.
Okay, thank you.
Yeah. Okay, thanks. Any other questions? Yeah, sure.
In the mammalian olfactory system, even to the same odorant, the neural representation is changing over time. It's called drift. Is there any kind something like that in the Drosophila?
You know very well. Yes, the representational drift is a relatively hot area in many modalities and so on. Yes, so it happens in the piriform cortex. We have an unpublished data, but I can see it here. Yeah, it happens in the mushroom body as well. It's very interesting. Yeah, I can. Yeah, let me talk to you later, and I can talk to hopefully everyone in a year or two.
Yes. I'm looking forward to your paper.
Yeah. Okay, another question. Is the 3-layer olfactory circuit in the fly you described here conserved in the mice? Yes. Okay. Mouse, I told you that it's quite similar. So the counterpart of the projection neurons are the mitral tufted cells. And then the counterpart of the canyon cells are pyramidal neurons in the piriform cortex. Now, what happens downstream of the pyramidal neurons is not well described yet. So, and also we don't know whether they receive neuromodulatory input for synaptic plasticity or not. So I must say that less is understood in mice. There may be similar plasticity happening. In that case, it becomes quite similar. Okay. All right, so let me move on and then finish the last part. Okay. Good. We are not so behind in time. All right, so the next question is, how does the brain compute the value of odors? So far, the entire field has focused more on the computation of the identity of odors or identity of odor groups and so on. But in the end, what matters the most for us is not to just discriminate the smell of an orange from the smell of an apple, but to tell whether that smell has a high value or a low value, whether it leads to an edible energy source or inedible rotten fruits or a scary bear trying to attack us and so on. So odors are intrinsically associated with the positive values or negative values. And we can perceive this value without any learning. Even if, when you smell the bad smell for the first time, you do perceive it as a negative smell, right? So it's a hardwired property, and it's critical for survival. So this section is about understanding the computation and mechanism underlying innate value computation. Okay, so how do we assess the value for flies? To do that, we built a virtual flight arena for the fly. So it looks like this. If we zoom in to the center, it looks like this. So the fly is flying in the middle of the arena, sandwiched by a pair of microphone, listening to the left wing beat and the right wing beat. Odor is applied from head on, and the visual feedback is given with the LED. Okay, so this is a summary cartoon. By hearing the sound, the frequency of the sound, we can tell how fast the fly is trying to fly. And also by calculating the difference between the sound level of the left and the right microphone, we can tell to which way the fly is trying to turn, even though the fly is fixed. Okay, and we do use camera to directly image the fly, but we want to update this virtual environment as quickly as possible. And doing image analysis takes more time because it's multidimensional, but the sound is one-dimensional, it's light. So we utilize the sound signal to update the environment every 5 milliseconds, depending on the turning behavior of the fly. And this is the raw data. The top two are the sound data. So if you look at the frequency, at one point, the fly decides to fly faster. If you look at the amplitude at the beginning, it's different. So the fly is trying to turn, but at this time point, the fly decides to fly straight. And the last row is a wing, the angle of the edge detected by the algorithm from the video data. And I hope you can appreciate that this sound data and the wing angle data is highly correlated. So from this, we thought that, okay, we can rely on the sound data to run this closed loop system. The task we asked the fly to do is quite simple. Make turns depending on the value of the odor. So to mimic the natural statistics of the odor plume found in the environment, we applied an odor whenever the fly is flying within the 45 degrees regions. Okay? So if the fly likes the odor, it will try to fly straight, hoping that the food is there. But if the fly dislikes the odor, it can make a rapid turn and escape to the air zone. This slide shows an example flight trajectory of a single fly responding to 3 different odors. Because we have applied an odor for 15 times, 15 traces are superimposed. X-axis is time, and Y-axis is the virtual 360 degrees heading direction. Okay. So if we look at the response to the air, just as we do random walks, flies would make random turns from time to time. But when we apply apple cider vinegar, which we know is attractive for fruit flies, they fly more straight. And if we apply 2-methylphenol, which is utilized as a disinfectant at a hospital, they make a rapid turn within 200 milliseconds or so, escape into the air zone, and then fly straight again. So just like us, they like the smell of a fruit and dislikes the smell of a hospital. Now, there are many ways to characterize this behavior, but here we simply calculated the proportion of time spent inside the odor plume and defined that as a value index. And we can quantify the value of the different odors using this simple calculation. And so we are ready with this odor set, ranging from very aversive to very attractive. Now, can we decode the odor value from neural activity and predict the animal's behavior? If we could do that, then we can say that we understood the relationship between the neural activity and the odor value computation or odor value dependent behavior. And that's one of the aims that neuroscientists aim. Okay. So how do we create a model that links the neural activity and the behavior? Well, we recorded from projection neurons in 37 glomeruli. So as an independent variable, we have projection neuron activity, okay? And the target is the behavior value index in this case. What is the simplest model that connects these two variables? The simplest model is always a linear model. So we first made a model that does the weighted summation of PN activity input, hoping that it will explain the behavior. By the way, a rule of thumb to generate a good model is to keep it as simple as possible, but at the same time have an ability to predict the responses to novel input. Okay? So the complex models are not necessarily a better model. And that's why we started from a linear model. Okay, we trained the model. So training means defining a proper synaptic weight that leads to the explanation of the value index. Okay, and along the y-axis, I'm plotting the actual data, the actual value index, and the y-axis is the model output. Using the training data, we were able to explain the existing data pretty well. But by inputting the order responses of projection neurons to an order that was never used to train the model failed quite miserably in predicting because the responses are everywhere with an R squared of close to 0. So the prediction doesn't match the actual data. Okay, so then we had to modify this model in some ways, and the question is why we needed to modify and how to modify. For the Y part, we thought that maybe flies adapted to the 6 orders that we applied during the experiment. To utilize the time effectively, we did not apply just a single order, but we applied 6 orders in a random order. And animals, including us, are really good at adapting to the statistics of the environment. So maybe we needed to incorporate this olfactory adaptation into account. To do that, we inserted this normalization step, which is to subtract the mean response to 6 orders and the division by the standard deviation of the responses to 6 different orders. Well, why we did this is because the vision scientists and the auditory scientists told us that this simple z-score calculation like normalization worked well to explain the neuronal adaptation to visual stimuli and the auditory stimuli. So we borrowed that idea into our model. So now we call this model a normalization model, and then found out that the prediction became better. Well, so it's biology, it's never perfect. And also we are recording only from 37 glomeruli and not 50 glomeruli. So the results shouldn't be perfect anyway. But so we trained the model using individual odorants and tested with mixtures that were never used to train the model. And the quality, accuracy of prediction was significantly good compared to the null hypothesis. And then we further applied orders at the different concentrations. And again, the model was able to predict the behavior pretty well. So with this model, we were able to read the fly's mind with respect to olfactory perception. How does the weight distribution look like? How do individual glomeruli contribute to the final behavior? To understand that, we looked at the distribution of weights and found that the weights are shared among a large number of glomeruli, and half of the glomeruli contributed to attraction and half of the glomeruli contributed to aversion. So they seem to be employing democracy to decide what is the best responses to each order. Now, so far, we have explained the model that captures the correlation between neural activity and behavior, and not causality. And it's important to understand the causality. So to test that, we did two different experiments. One is to block the neural activity or the neural input, so the loss of function experiments. A toxin called tetanus toxin was expressed in 1 or 3 types of olfactory receptor neurons to block synaptic inputs to glomeruli. And then compared the value index, the behavior in the control fly and the TNT expressing fly. And we block the input to individual different glomeruli or a combination of 3 glomeruli and so on. Okay. And we found out that the responses deviated in the TNT expressing flies from the control data in a direction that was predicted by the model. And I'm showing the prediction R squared, which are significant. The projection neuron activity in glomeruli are indeed causally related to the behavior. We did a reverse experiments, now doing a gain of function experiment, activating a glomerulus that is usually not activated by a particular odor using optogenetic approach. We express red sensitive crimson in a particular glomerulus, and then applied an odor together with a red light. And then we calculated how much the value index differed with or without light. Okay? And we found that co-application of laser and odor changed the fly's behavior, again, in a direction predicted by the model. So it was good. And then the very, very final section is the favorite of mine, which is that the model predicts that the relative value can change depending on the olfactory context. So there was an additional qualitatively different prediction made by the model. And I will explain slowly what I mean by the change in relative value. Okay, so on this slide, x-axis is the attractiveness of an odor. So you like the smell of a lemon a little bit more than the smell of a celery, okay? And those two odors are identical in 2 different contexts, context A and context B. You create differential context by bringing in different odors, okay? So these 4 odors together form a context A, and these 4 odors together form a context B. Now, the equation tells us that depending on the odor context, the smell of a lemon will become much, much more attractive than the smell of a celery. And in another case, the value can even switch. This is quite surprising. But the equation tells us that this will happen. So we asked the fly whether a fly's perception changes or not. We prepared 6 odors forming context A and another 6 odors forming context B. But the 2 odors were kept identical. So in the previous cartoon, the smell of a lemon and the smell of a celery. Now we are interested in the relative difference in the odor valence or odor value, okay? And whether this difference changes between context A and context B, okay? So here I'm plotting the difference between the value index of odor 2 and odor 1 for a particular odor pair, okay? Now, in context A, the fly said, oh, I dislike the odor 2 more. But in context B, the preference actually reversed. I now dislike odor 1 more. And this happened not only in the one particular odor pair, but in other odor pairs. And importantly, this fly's actual behavior was properly predicted by the model. So whenever a model said that the relative value should change, then the fly changed its preference. And whenever the model said, oh, in this particular odor pair, the preference doesn't change. And in that case, the fly didn't change the behavior. So like the physics experiment where the theory is built first and the experiment follows, in the biology experiments, we were follow the same sequence. And this is quite unusual. And that's why I felt satisfying by seeing this result. And the good thing is that if this approach is successful, then we can form a better experiment to do subsequently. And also trying to come up with a unifying computation that happens in the brain. So to summarize, we have constructed a decoding model that can predict the animal's perceptual decisions in response to a diverse set of odors. Behavior is dictated not by a small subset, but rather by many glomeruli, each of which having specific contribution to attraction or aversion. We found that the relative odor value can switch depending on the context, as predicted by the normalization model. This suggests that the olfactory system can adapt to its environment similarly as visual and auditory systems. So with that, I would like to thank my lab members, and I'm happy to take any questions. Yes.
So thank you for the lecture. And do you mind telling me a bit more about how they upgrade the order value by past experience? Like you just mentioned, the relative value can change depending on the context. But what about more like longer period of time? How does it affect?
You're interested in the time course.
Yeah.
And I guess also the mechanism, a little bit more detailed mechanism as well. I didn't have time to explain the mechanism.
Oh, like I feel like environmental stimulus can affect the relative value of others. Like you said, whatever more, I don't know what you said.
Okay, you're interested in the time course. Yeah. Yes.
Last.
Yes. Okay, good question. So this switch in relative value happened within 10 minutes or so. So one may think that's pretty quick, or one may think that's pretty slow. We need to think about the plasticity underlying these changes, right? And that plasticity should have a time constant of about 10 minutes or 15 minutes or so. Now, what could be the mechanism that can give rise to that time scale? I still don't know the answer, and we are trying to figure out the mechanism underlying this plasticity. It could be neuromodulatory effect that kick in slowly, or synaptic plasticity that doesn't happen rapidly like LTP, LTD, but non-canonical type of synaptic plasticity that happens on a longer time scale. For example, at the projection neuron to the third order neuron synapses and so on. So there are many possibilities, and we need to examine that, but we don't have the answer yet. Yeah.
So like, is it even possible that you manipulate that specific preferences of others to, is it possible that you make aversive odor to more Like,
Yeah, we can do that because we have the equation. And according to the equation, we made a prediction, right? So using optogenetics, we may be able to create a synthetic odor, synthetic background odor, I should say, or the context forming odor that induces this changes in relative value. And we do not necessarily need to rely on the actual odors, but with the optogenetics, we can activate specific sets of glomeruli. And then I believe that theoretically, in principle, it's possible to induce this plasticity.
then sorry, are there certain period where you can change it more easily? Like, are there any critical period?
I see. Developmentally, I
Yeah.
mean. We haven't tested at different ages, so I don't know the answer to that question. Yeah. Okay, so I'd be happy to answer any questions anytime. So you are feel free to email me if you came up with more questions later on. Also, I'd be happy to show you around our lab space and see the actual setup.